So this is the last installment for the May-June 2018 paper 11. We are on question 36, and there's only 40 questions in the paper, so we should be able to finish them in this lesson. So question 36, we've got a whole lot of information, so we need to read very carefully. The diagram shows one way of testing the effect of an antibiotic on bacteria. So we have a Petri dish with nutrient agar containing bacteria. We have a disc of filter paper soaked in antibiotic, and we have colonies of bacteria. Okay, so this is grown for 5 days. And the diameter of the zone is measured every day for 5 days. So is the antibiotic killing off any bacteria in this region where it's close to the antibiotic on the filter paper? Okay, so the table shows the results of testing 5 different types of bacteria. So we have 5 types of bacteria, days 1 through 5. They tell us something very important here. They tell us that zones of less than 13 millimeters show the presence of resistant bacteria. So we need to go and we need to have a look and see, where do we see less than 13 millimeters? And here, day 3, they're cropping up, day 2 cropping up, 13.2, not quite less than 13. So then 9, 11, 8, and 2, and 10, and 2. But bacteria in 1 and bacteria 5, they don't show any resistance. So then we have to look at their statements and we have to decide, do the data here support this statement or not? Part A, all the types of bacteria become resistant to antibiotics over time. That's far too general. We are testing this one antibiotic and we are testing only 5 types of bacteria. So there's not a chance we can make a statement like that. Only types 2, 3, and 4 of the bacteria show resistance to the antibiotic. Well, we've only circled these less than 13 showing resistance in 2, 3, and 4. So this one we agree with. But let's confirm with the other 2. The antibiotic can be used to treat types 1 and 3 only. Well, 3 is resistant, so we can't be treating 3. 1, yeah, okay, 1 is not resistant, so we could potentially use it for 1. Type 5 of the bacteria can never become resistant to the antibiotic. Never say never. So it isn't resistant at this stage, but we don't know about it in the future. Our data does not support that. So we have confirmed the answer for this question is B. Question 37, another long and informative question. Some animals have genes that code for small peptides called catholycidins. However you pronounce it, doesn't matter as long as you can read it. These peptides kill a wide range of bacteria by attaching to lipids in bacterial membranes, so weakening or disrupting them. Okay, so it's a peptide that attaches to a lipid, and then the membrane is dysfunctional. Scientists have produced a synthetic version of the catholycidin that kills bacteria that are resistant to a number of antibiotics, such as tetracycline. So we've got our antibiotic-resistant bacteria, but now this peptide is not an antibiotic. Okay, so it's a peptide that can weaken the membrane, so we can still kill the bacteria. Which pair of statements explain how this synthetic catholycidin might help with the problem of antibiotic resistance? So we have to pick 2 answers out of here, and all our answers are 2 possibilities. So it is synthetic, so bacteria can never become resistant to it. Never say never again. Okay, and bacteria are constantly evolving, and you never know what's going to happen in their genetics. So maybe they actually then get a mutation that could alter their lipids. That means the catholycidin wouldn't be able to bind to it. So that one is not a good option. Option 2, it could be used instead of tetracycline, allowing tetracycline resistance to be reduced. Well, that's true, because it will kill the bacteria, even if they're tetracycline resistant. The only way a bacterium could develop resistance to it is by altering all the lipids in its membranes. That sounds a little bit too general, so I'm not too happy with that one. So let's check the 4th. They could be used to kill multi-drug resistant strains of bacteria for which there is no effective antibiotic. Well, that's true, because this is not an antibiotic that we're working with. So going back to 3 again, if we're looking at this, okay, the only way bacterium could develop resistance is by altering all the lipids in its membranes. Well, not really, because we've got a lot of variation going on, and there's far too much to consider under this. It's not a tight enough answer. So we're happy to cap that one out and go for 2 and 4. Question 38. Hybridomas are used as a basis for the production of large numbers of monoclonal antibodies. Which statement describes how hybridomas are made? So we don't look at the answers. We go back to hybridomas, and what are they? They are cells that produce antibodies and divide so that we can get even more antibodies. So antibodies are our B lymphocytes, and never stopping dividing is our cancer cells. So where do we find B lymphocytes with cancer cells? It's here in option B. Question 39. Which statement about the properties of the antigen binding sites in antibody molecules is correct? So if we think about our antibody molecule, we've got this sort of a structure. We've got heavy chains, we've got light chains, hinges, disulfide bridges. Okay, so antigen binding sites are on the light chains only. Nope, they're on both. Antigen binding sites have a hinge region to give flexibility for different antigens. No, the hinge region is very different. It's not for different antigens, it's for a different function of this molecule. Different antigens is about specificity and structure of that binding site. They have binding sites for receptors on phagocytes. No, the binding sites are all about the pathogen. They have variable amino acid sequences for different antigens. Yes, that is true. Same like enzymes have different active sites for different substrates. Our antibodies have different amino acid sequences, so they're going to correspond with different antigens. So 39 is D. And the final question in this paper, question 40. The statements describe ways in which different types of monoclonal antibodies can work. They can bind to proteins on cell surfaces and trigger the immune system. They can block molecules on cell surfaces that inhibit T cells. They can block cell signaling receptors that trigger cell division, and they can block cell signaling receptors that trigger the immune response. Which types of monoclonal antibody could be used to treat cancer? So in the context of cancer, would we want to be able to bind to proteins on cell surfaces and trigger the immune system? Yes, because then we could have our immune system actually attack those cancer cells. Would we want to block molecules on surfaces that inhibit T cells? Absolutely, we want to, because we want to stop them inhibiting. We want the T cells to work. Block cell signaling receptors that trigger cell division. Yes, because if we can stop cell division, then we slow down that growth of the tumor. Would we want to block cell signaling receptors that trigger the immune response? No, that's the opposite of the first one. Okay, we would want that immune response to be active if we're treating cancer. So we've come out with 1, 2, and 3, which corresponds to option B. And that then is the end of the paper. So when you're working through your multiple choice paper, you've got 40 questions and you've got 1 hour to do it in. Okay, so it is important that you work through systematically through the paper. You can always come back to questions that have confused you, and always make sure that you are scribbling on your questions, interact with the papers. Okay, so I had a question come through about statement 2 here, that it's a bit confusing. Okay, so let's say we have a cell surface and we have a molecule here that inhibits a T cell. In other words, it prevents the T cell from attacking this cell. Now, what we're saying is a monoclonal antibody can go and it can block that. Okay, so if you're blocking that T cell inhibition, so T cell inhibition, if we block this, then we have T cell functionality. And if we have T cell functionality, then we can attack that cancer cell. Does that make sense? Cool. All right. So multiple choice papers, very good practice for when you're working through your revision and making sure that you're comfortable, as they're going to cover everything in your syllabus. So the idea here is to work through some questions that deal with DNA, but in particular that have a piece of mutation in them, looking at how it can be asked in a question. So starting here, we have a question. It's question 5 from Winter 2012, paper 23. Question starts out with the role of the ribosome, then looks at DNA and goes through to then ask about mutation. So starting from the very beginning, describing the role of the ribosome in translation. So the big thing is, the ribosome, we're going to run through this one relatively quick so we can discuss when we get to the mutation ones and see if we have time for a few more questions as well. Okay, so our ribosome attaches to the mRNA. Within that ribosome then, we have 2 codons of the mRNA exposed. Okay, so the codons provide the code. The ribosome provides the attachment site then for the tRNA. So mRNA provides the code for the sequence of amino acids. The ribosome provides attachment sites for 2 corresponding tRNA with their specific amino acids. So we've been asked to describe here. So we're giving a little bit more than just attachment sites for tRNA. What's the importance of those attachment sites? Is that we have the mRNA and we have the complementary tRNA, that it's all sort of working together. Okay. So we have the attachment site and then we get that codon, anticodon binding within the ribosome. And that then means that our peptide bond can form. Our ribosome contains peptidyl transferase to catalyze the peptide bond formation. And with my big ugly online handwriting, I've run out a bit of space, but you can see with normal handwriting we'd be doing quite nicely. Okay, once we got that peptide bond formation, the important thing is the ribosome moves on 1 codon at a time. So it would have fitted beautifully into our allocated working space. So describing the role of the ribosome, what's happening, but with specific response to the pieces of the ribosome. What is happening exactly in that ribosome? Why is the ribosome important for translation? So figure 5.1 is a diagram of a section of mRNA showing the sequence of 3 of the codons, codons 1, 2, and 3. And we're going in this direction for polypeptide synthesis. State the base sequences of the tRNA anticodon complementary to codon 1 and the DNA sequence which coded for codon 3. So I'm just going to split them like this so that I can see them more easily. So the tRNA going to codon 1 would be G, G. I'm saying G's are writing C's. G, G, C. So here we would write G, G, C. And the DNA that made codon 3. So opposite of G, we would have a C. Opposite of A, we'd have a T. Opposite of U, we would have had an A. So our DNA would have been C, T, A. So now the focus of this, the 3 codons in figure 5.1 are near the start of the sequence coding for a protein. Explain the consequence of a mutation which deletes the U from codon 2. So this one here would go away, completed deletion, loss of it. So what we need to think about is what would then happen. The consequences, so starting from that mutation, what would happen? Well, first off, we still need 3 to get to an amino acid, so now we'd be going AAG instead of UAA. So the amino acid coded for by codon 2 is changed. This is an example of a frame shift mutation. So everything is going to move, which means that every amino acid downstream is going to be different. So every subsequent codon is different. So the amino acid sequence changes. When are we changing this? Remember, we have stop codons. So if we change the frame in which we are reading this code, we could all of a sudden have a stop in the middle of our protein. So what's this going to do? We're changing amino acids. We're potentially stopping early. We are changing the primary structure of the protein. If we change the primary structure, we change the secondary and the tertiary structure of this protein as well. And that makes it dysfunctional protein. So the ultimate consequence of this mutation is a dysfunctional protein. So we're going to change the primary, secondary, and tertiary structure of the protein, leading to a dysfunctional protein. So in this question, they're not asking us how did this mutation happen. They're asking what's the consequence. So we've got this mutation, we've changed the second codon. From here, what is going to be different moving forward? Are you happy with this question? Okay, it seems like everybody's happy, so we shall move on to another mutation-based question. This one is now by June 2015, paper 23, and we're working on question 2. So again, we set up with a bit of DNA stuff, and then we get into what is meant by a June mutation, and then how can that affect, in this case, cell cycle. So DNA replication is an important event in the cell cycle, very important. State when during the cell cycle DNA replication occurs. So it is in interphase. It is late in interphase. Figure 2.1 shows pairing between two bases, X and Y, in a DNA molecule. Name the type of bond shown by the dotted lines between the bases. How do we show with a dotted line? Which bond is that? It's always the same thing because it's not as strong as a normal full bond. It is a weaker bond. It is our hydrogen bond. And then they ask us to state which base, X or Y, is a pyrimidine, and explain your answer. So remember, you've got your pyrimidine and you've got your purine. Now the thing is that their sizes are back to front with their names. The pyrimidine is the smaller one and the purine is the bigger one. So Y, single ring structure, is a pyrimidine. So Y is a pyrimidine because it has a single ring structure. And here comes our mutation question. The compound benzopyrene found in tar from tobacco smoke can become chemically changed in cells and interferes with DNA replication, causing gene mutations. State what is meant by the term gene mutation. So state, but for 2 marks. So we really need to be thorough in what we're saying is a gene mutation. So at the very basic of the beginning, it's a change in that nucleotide sequence. So change in the nucleotide sequence of DNA. Okay, so that then leads to changing of the mRNA, and that then leads to changing of protein. We've got enough space to put those both in. But let's be a little bit more examples, potentially, of mutations. So we could have, possible, no, not most, but possible is not the right word. We can have mutation types. We can have the deletion of a base. We can have the addition of a base. Or we can have the substitution of a base. So remember, deletion and addition, these are our frame shift mutations. Substitution only affects the single codon. Well, the single triplet in the DNA, single codon in the mRNA. So we have different types of mutation. This alters mRNA, which is formed in transcription. And this then changes primary structure of the protein. So our main focus of our answer is these first 2 things, where what is the gene mutation? But because we've got 2 marks, we've got a bit more space, we're taking this a little bit further into what it is that's happening as a sort of result of the mutation. Last piece of this question. Mutations that occur in dividing cells of the gas exchange system may result in lung cancer. Suggest the differences in the cell cycle of a cancer cell compared with that of a normal cell of the same type. So it's a suggest question, which means you've got to think about what you know and then you've got to apply it in this situation. So what is the thing about cancer? It's about that uncontrolled mitosis, that lack of regulation. So we can start there. There is no regulation of the cell cycle. Because of that, you would have uncontrolled cell division. Now, what's going to be the implication of that on interphase? We would expect that if we go and divide, that interphase is going to be shorter. So we're going to have uncontrolled cell division. We're going to have shorter interphase, and nice one to add, of course, we're not going to differentiate. There's going to be no specialization of cells. So no differentiation of cell types. So there's quite a lot of different ways. I mean, I've got more questions here as well, but we run out of time to go through them at this stage in the way we can ask about mutation. So it's really important that you look at the question very specifically and figure out what is it that they are precisely asking you with regards to mutation in this case for this question.